phylogenetics. We looked at diagnosis of human genetic diseases. We mentioned a few examples. All right. Then we look at DNA fingerprinting, where forensic science plays a key role in trying to pin a suspect to a crime scene. And we look at human gene therapy, where we have a defective gene causes a particular disease. If that gene could be replaced with the wild type, then you could actually solve that problem, that disease, using gene therapy. We looked at the Human Genome Project. We also look at some commercial products that were developed using recombinant DNA technology. All right, we have the TPA, we have HGH, we have insulin, etc. And then we look at genetic engineering of plants and animals. All right. And so now we're going to talk about genomics. What is genomics? I want you to pay keen attention that genomics is different from genetics. You all know already the definition of genetics. All right, so it has to do with the study of genes. We're talking about the function of genes, the structure of genes, and how these genes are passed from one generation to the next. That is genetics. Now we will look at genomics. All right, they're different. But in genomics, we can locate a gene on a chromosome in a subregion of a chromosome and finally its precise location in the chromosome's DNA sequence. The DNA sequence level requires knowing the entire sequence of the genome, something that was once out of our reach technologically. But we all know that you can do genome sequencing, whereby you sequence the entire genome of an organism. What does that mean? It layouts the order of these nucleotides, so the A, T, C, G, and so on, down the road of the entire genome, which means that the entire genetic material that is in the cells of these organisms. All right. Maps of genomes are constructed at different levels of resolution and using different kinds of information. We can distinguish between genetic maps and physical maps. All right. Genetic maps are abstract maps that place the relative location of genes on chromosomes. What do you call the location of genes? What do you refer to the location of genes? What's the term for the location of genes on a chromosome? Anybody knows? Come on, you all fall asleep so early, man. We're just, what, 5 minutes into the class and you drop asleep already? Some people, they just wake up, sign in, and go back to sleep. Everything is done right in their beds.
The nucleus, sir.
The what? Nucleus? No, the nucleus is where you would find the genetic material in eukaryotic cells, right? But I'm talking about where a gene is located on the chromosome. So you have an entire chromosome. All right, so you have a chromosome here. And I say, okay, a gene is located right here. What do you call that position? So this would be the locus, all right? Locus is the position of gene. Plural would be loci, all right? Okay? So the relative location of genes on chromosomes based on recombination frequency. We talked about this in our previous class, right? Using recombination frequency in order to determine distance between genes, all right? Physical maps use landmarks within DNA sequences, ranging from restriction sites to the ultimate level of details, the actual DNA sequence. So DNA sequencing plays a key role in building physical maps. So if we look at this diagram here, we talk about types of genetic mapping. You have a genetic linkage map and physical maps. In the case of genetic linkage maps, arrangement of genes and genetic markers on the chromosomes, that is this one at the top here. So it gives an idea more or less where these genes are located. And of course, that can be done using recombinant frequency All right, so you obtain your F2 generation, and then you calculate how many recombinants you obtain, and then you calculate your recombinant frequency, and that could determine distance between genes on a genetic map. Physical mapping, on the other hand, provide exact physical distance between the genes located on a chromosome. And of course, it is ideally measured in nucleotide bases. So when you look at this map at the bottom, the physical map, it can sequence. DNA sequencing plays a key role in terms of developing physical maps, all right? So when you look at this portion right there, it gives you the DNA sequence, all right? And it is in bases, okay? If you notice the genetic map, we're talking about centimorgans, all right? Which is the distance on that chromosome, okay? Cytogenetic map, all right? This is an overall diagram of the chromosome. Here you have the centromere. And did we talk about long arm and short arm of chromosome? What letters are assigned to them? Did we? What do you call the long arm of a chromosome? It's a letter. Did we?
I remember to talk about that, sir.
We don't remember talking about that. Okay, so you have a chromosome here. So this is the centromere, all right? This is the short arm, and so this is called the P arm, all right? So the short arm is called the P arm, and the long arm is called the Q arm, okay? So this right here, this just gives you an idea of the chromosome itself, where the centromere is located, the long arm, the short arm, and of course, subregions of this chromosome. Right, so that would be the cytogenetic map. So the cytogenetic map doesn't give you much details in terms of where genes are located on the chromosome. Proteomics, all right? So from the term, you know it has to do something with protein. So what is proteomics? There's the study of the proteome, and the proteome is all of the proteins encoded by the genome. So remember we talk about DNA replication, then transcription to RNA, and from RNA, translation to proteins, all right? So remember that the entire genome will code for a group of proteins. Well, the study of that entire protein, all the proteins that are produced by the genome, is proteomics, all right? So understanding the proteome for even a single cell will be a much more difficult task than determining the sequence of a genome. Because a single gene can produce more than one protein by alternative splicing. The first step is to characterize the transcriptome, and the transcriptome is all of the RNA that is present in a cell or tissue. So when you study all the messenger RNA that are produced from transcription, all of those, that is called transcriptome. So the transcriptome consists of all the messenger RNA produced, okay? Proteomics moves from genes to proteins, all right? That's the central dogma of molecular biology. Proteomics characterizes all of the proteins produced by a cell. The transcriptome is all the messenger RNA present in a cell at a specific time. Why is this important at a specific time? Because we said that some genes are dormant and they're not activated until certain developmental stages of an organism. All right? So there might be some stages in an organism where specific messenger RNAs are not present. And hence the reason say at a specific time. Protein microarrays can identify and characterize large number of proteins. So what are the applications of genomics? Why study the entire genome of an organism? Genomics can help to identify infectious diseases. All right. So genomics can help identify naturally occurring and intentional outbreaks of infectious diseases and tracing of disease strains. This right here, tracing of disease strains. You hear about COVID-19 and different variants. All right, from the time COVID broke out, we have had so many different variants of COVID-19. How are they able to identify these variants? Well, through genomics. You have to sequence the genome of the virus. All right. So when you sequence the genome of that virus, and the SARS-CoV-2 virus is approximately 30 kilobase pairs. 30 kilobase pairs. So we're talking about 30,000 bases. That is the size more or less of that SARS-CoV-2. So from the time that broke, that genome of that virus was sequenced. So they know exactly the sequence of these bases. And so when there are changes in these bases, there is where you have new variants. And these new variants come with all different kind of things. Probably they're more infectious, they're more deadly, they're more, it varies. So from the fact that you can change a single, or it changes spontaneously, all right? It changes on its own. So you have changes in these bases in the genome, there is where you have a new variant. And the only way you can identify a new variant is by sequencing the genome of this in this particular case, the virus. And so you can compare the original DNA sequence with the new strains and see exactly where the changes took place. All right, so genomics can help identify these infectious diseases and of course identify these variants as well. Genomics can help improve agricultural crops. We talked about this in the application of recombinant DNA technology. So it can potentially increase the nutritional value of crops and alter their responses to environmental stresses, potentially helping to feed a growing population. So for you to be able to use genetic engineering and develop GMOs, genetically modified crops, you have to have an understanding of the genome of that organism. Where is it that you want to insert a gene that would give that plant more resistance to a particular pest or disease? Where is it that you want to insert that gene for that plant to give you higher yields? So you have to have an understanding of the genome of the organism when you're using recombinant DNA technology to develop genetically modified organism, okay? Third application is genomics raises ethical issues over ownership of genomic information. So questions regarding profit and ownership of genomic data provide ongoing challenges for the ethical use of scientific knowledge. And this is a ethical issue that is debatable. If I sequence the genome of a particular organism, does that make me the owner of that particular genome? Big question mark over there and big debate going on there. So I might invest resources for me to be able to sequence the genome of a particular organism, but at the end of the day, that organism is out there. Other people can sequence it as well. All right, so you can't say, well, because I was the first to sequence the genome of a particular organism, now I have ownership of that. And you want to make profit off that. And anybody who wants to use that particular information or that data, you need to pay me some kind of royalties or some kind of monies for them to use it. That's not so. It's out there and it's a big debate going on, right? All right, I will show you guys a couple videos right now. I think the first one has to do with genomics, and then the other one will talk about proteomics. So these are two. Go ahead, Delvin. Yes, exactly. You patent that information, that data, and you call it yours. So anybody want to use it must pay you. Well, there is where it goes, because DNA sequencing is becoming more regular. It's easier now than before. So somebody else can go out there and sequence that as well. But the whole issue in terms of from a legal standpoint, if you already have the patent for something, then that might prohibit that person from going and doing it, or even do it and call it their own. All right. So you can see where not only ethical issues come to play here, but legal issues as well. All right. Somebody who say, all right, I did it first, I patent it, it's mine. So nobody else can do it. So that would have to be something to be determined in a court of law. All right. And something similar happens to GMOs, but in terms of GMOs, it's a little bit different. When I know that when someone develops a genetically modified organism like BT corn, they patent that. That is patented, so that is theirs. So what they do is they, and it is usually these large seed companies that are engaged in genetically modified organisms or the production of these organisms, and they're the ones who commercialize it thereafter. All right. So, and if anybody else out there who want to grow these GMOs, then at the end of the day, the same company will benefit because they have to come to the company to purchase these plants. All right. But all these major companies who have developed genetically modified organisms, trust me, they patent their work and their GMOs. That is patented. All right, so the first video will talk about genomics, and then the second video will talk about proteomics. All right.
What is genomics? The first step to understanding genomics is to ask, what is a genome? A genome is the set of instructions needed to create an organism. Genomes are stored in cells as DNA. DNA is short for deoxyribonucleic acid, an amazing molecule that stores information using a special code. You can think of a genome as a coded cookbook to build and grow an organism. For example, you. The human genome contains about 23,000 recipes that code for different proteins. Each recipe or functional unit of DNA is called a gene. But only about 2% codes for protein. The rest is non-coding DNA with regulatory or other yet unknown functions. The study of how DNA works is called genetics. The methodologies that define genomics allow us to see a bigger picture, a panoramic view of the entire DNA landscape. Not only how DNA works, but how the whole genome interacts with its environment, from the cell that houses it to factors outside the organism. The study of the genome and its environment is known as genomics. A famous example of the power of genomics is the Human Genome Project. The goal of the project was to read the entire human genome. That's enough information to fill not one, but 400 standard English dictionaries. As the project progressed, researchers needed to create new tools to analyze and share this massive amount of data. This, in turn, spurred an explosion of further research. By its publication in 2001, the Human Genome Project had forever changed the way we think about life sciences research. How genomics is improving lives. 2 Made in Canada examples. Example 1, copy number variation. Until recently, it was widely accepted that the human genome contains 2 copies of any given segment of DNA. By looking beyond the single gene level, genomics revealed a different picture. We now know that large segments of DNA can actually be duplicated or deleted in the genome. These copy number variations, or CNVs, contribute to our uniqueness, including susceptibility to some disorders. By examining CNVs, Canadian researchers are learning more about how genomics can improve human health and are sharing their results with the world using an online database. Example 2. Combating the mountain pine beetle. In Western Canada, the mountain pine beetle has destroyed millions of trees. The beetle carries a fungus that helps it to overcome the tree's defenses. Overrun with invaders, a tree will eventually die. Researchers are currently studying the interaction between these three organisms at the genome level to uncover which characteristics of the beetle allow it to carry the fungus, and under what conditions trees are able to fight off invasion. The results will lead to forestry management strategies that can save our pine trees. The speed of genomics. The Human Genome Project took 13 years and over $3 billion to complete. Since 2001, individuals have had their genomes read in a fraction of the time for a fraction of the cost. Soon, you'll be able to have your genome read quickly and cheaply. Expanding knowledge. Today, the genomes of thousands of different animals and plants and bugs have been read. By comparing genomes, we can see just how alike different species are, not only today, but through evolutionary history. Open access. The ever-growing list of freely available genomics resources is accessible to everyone, including you. Open access to such a wealth of knowledge allows many positive applications which impact our lives, our health, and our environment. Genomics is holistic in scope. It studies all the genetic information in an organism and how that genome interacts with its environment. Genomics is high throughput. It reads and analyzes large amounts of DNA information quickly and efficiently. Genomics is resource generating. These resources further enable research and discovery. Genomics is holistic, high throughput, resource generating.
I got loyalty inside my DNA. Quarter piece, got war and peace inside my DNA. I got power, poison, pain, and joy inside my DNA. I got hustle, though, ambition, flow inside my DNA.
All right, let's kind of recap where we are so far in the course. We looked at mass spectrometers, different mass analyzers, different ionization sources, and basically we understand that this machine is very complicated, but also very versatile. There's a lot of different things you can do with mass spectrometry. So I could talk forever about different ways of using mass spectrometry, but I decided to use what is kind of near and dear to me when it comes to proteomics analysis. That's sort of my thing. That's what I do. So that's what we're going to be talking about in the next couple of videos. But in this particular video, you're not going to hear really anything about mass spectrometry. I kind of have to set the stage. So when it comes to doing those types of different MS analysis, there's a lot of different versions of it, but we've got to understand what we're studying first. So this is a recap of proteomics. What is it all about? Okay, I don't know what everybody's background is in terms of biology, so let's just start simple. DNA, RNA, protein, that's about it. That's like kind of all we need to know. In other words, we understand that DNA is kind of what makes us us, right? It's the code. It's the thing that tells us who we are. But when it comes to the sort of evolution, we're talking DNA, which makes RNA, which ultimately makes protein. So DNA doesn't do anything. And the good analogy of it is to think that DNA is like a blueprint. It's just like a set of instructions to decide what to do with, an organism. Proteins are what actually carries out all the tasks. Proteins do a lot of different things. They can be thought of as like enzymes that assist in chemical reactions, but they could be structural as well. So the proteins that make up your your hair and fingernail, that's just protein, right? There's instructional proteins, there's proteins designed to communicate with one another, there's receptors, things to carry things. There's like so many different varieties of proteins. DNA doesn't do any of that, but DNA codes for all of the proteins that carry out the functions of our body. All right, without getting too technical here, we can think about the chemical composition of DNA versus proteins. So DNA carries with it 4 different bases. Let's just say A, T, C, and G, whereas the proteins are made of amino acids. And we usually think of these 20, let's say, common amino acids. Yeah, there are a couple of other ones out there. But we've got 4 letters that make up DNA. We have 20 different varieties of chemicals that make up proteins. So how do we go from 4 to 20? Well, it's really the same way that how we take an alphabet of 26 letters and turn it into like thousands, 100,000s of words. So what you do is you string together 3 letters of amino acids. We call that a codon. And within these enzymes and transporter, the tRNA is what can read the DNA. So every 3 letters of DNA will convert itself into a specific amino acid. And when it comes to building these things up, that's all you need to know. So you see the letter codes there. No, it's not like I'm going to ever ask you to do that, to memorize these things, but like some people know them off by heart. So when it comes to DNA sequencing, all we're really interested in is determining that letter, A, T, C, G, the order of those particular bases. And yeah, in the grand scheme of things, if we know that codon sequence, if we know our DNA, then we pretty much understand what the protein is that's going to be coded from that. When it comes to DNA sequencing, there are today a lot of tools available to us, but it all started back here with Fred Sanger. So he won a Nobel Prize in 1980 for development of a technique called Sanger sequencing to be able to sequence DNA. You know what? He actually came up with another technique to sequence proteins as well. So he won 2 Nobel Prizes. Like, that's not a bad career. Anyway, what Dr. Sanger is holding in his hand is like one of those X-ray film kind of images, and there's a bunch of blotches on that, but you can read from top to bottom along 4 of the ladders in there, and you can actually tell what the sequence is. It gets a little bit more colorful, or actually literally more colorful, when it comes to like tagging the DNA with different fluorophores, and you can do separation from that. Ultimately, these are the techniques that led to the DNA sequencing of the entire human genome. So the Human Genome Project was a story that took actually not that long. When they came to thinking about how much it would take, it was about a decade worth of work, but people thought it would take a lot longer than that. The reason why it came to be quicker was because new, faster, more improved DNA sequencing technology continued to be developed. Nowadays, you want your entire human genome sequenced, you can do that. Like you can pay to get your DNA sequenced, and it's like $1,000 project. So this is one of those very common plots that we look at that looks at the price of sequencing a single base pair of DNA, and you can see that it's going down way beyond exponentially. Like it's hit any prediction and then some. And oh yeah, in terms of how it happens, well, I guess the secret really comes down to the fact that DNA is designed to copy itself. So if you can take advantage of that, as you're seeing in this video, then that's all it takes. So I guess what I'm trying to say here is that DNA sequencing, by today's standards, is a walk in the park. It's a very simple thing to do. We have the technology. We can do it quick. We can do it cheap.
We have the technology. We have the capability to make the world's first bionic man.
So it does beg the question, if we know the sequence of DNA, and DNA codes for protein, then what's the point of trying to study proteins directly? Why don't we just focus on the DNA and then we'll have our answer from there? Well, unfortunately, there are a few very critical questions that we're lacking when it comes to DNA sequencing. So a good example is right here. If DNA codes for RNA, which codes for protein, what's the connection? I mean, every single cell in our body has a single copy of DNA, but we have multiple proteins, multiple replicates of the same protein within every cell or outside the cell. So what's the number? What's the connection? Is it, how do we know how many protein molecules to synthesize? Well, that's something that's not coded directly at the DNA level. and you're better off to just sort of assay the protein to figure out what the concentration is. So here's a nice example of this type of study. Now, studies of this nature are absolutely massive. So this one here was a large consortium of scientists that were looking to study Alzheimer's disease and understand if there was a genetic link or a protein link. In other words, is there something going on in the body that is indicative of having Alzheimer's? Ultimately, does that mean, can we predict it before the onset? Can we find treatments for it? These are some of the questions when it comes to studying proteome, be able to understand what we're dealing with. So what did the study do? Well, it studied different brain samples. There's 453 unique brains, but actually it was 2,000 different samples. And okay, I'll admit, I didn't actually read this paper. There's like 160 pages of supplemental material, figure upon figure, data galore. This is just one figure from the entire study. To summarize it, basically they found the characteristic proteins involved in glycolysis, involved in generating energy for our brains, were disrupted when it came to Alzheimer's disease. So I don't know, maybe that's something that has to do with ultimately finding a way to combat this disease. Now, it's not just the abundance. So that study on Alzheimer's was looking at the fact that some proteins, there was more of it or less of it than what we would normally expect. But another key limitations of genome analysis is that it might tell you what the protein is, but it doesn't tell you what flavor, what form that the protein is. So the term proteoform is a new term that we came up with to try to decipher what one protein might be different from another. I'll give you a good example, blood type. So with blood type, we have type A, type B, type O. These are all the same protein, but they have different sugar units attached to them. So the protein itself is the same, but all of the things that come along attached to that protein are quite different, which is what you're seeing over here. And there's all kinds of different modifications. We refer to them as post-translational modifications. In other words, they're things that decorate the protein after they've been made. But those different decorations are key to deciding whether the protein is functionally active or whether it's sort of in a temporary state. Now here's a great example. So I mentioned the idea that DNA doesn't directly code for how many proteins are going to produce from it, but in a way it is responsible as well because it produces the proteins that are responsible for its own making. It's chicken and the egg kind of story here. Anyway, what you're looking at here are histones, and DNA will wrap itself around these protein molecules. And whether the DNA is coiled within that histone or loosely unraveled is one of the key mechanisms that decide whether the DNA is going to be transcribed or whether it will just sort of stay temporarily stored. So these histone proteins, well, a histone is a histone. The DNA, the amino acid sequence of the histone remains constant. But what does change is the modifications. And the modification levels on histones are absolutely insane. And if you think about the different combinations, let's say one histone protein has, 10 different sites that can be modified, each of them in 3 different ways. Think of the number. I'd have to sort of throw a number up on the screen to be able to figure out what that is. So yeah, you can kind of see that proteomics has a lot of questions to answer. So one of them being like how many proteins there are, being able to quantify the protein abundance. Another one would come down to those modifications, like whether they're there or they're not. But I guess the fundamental questions of proteomics relates to what is the protein in the first place? So when it comes to studying the structure of a protein, the first thing that we look at is simply the primary amino acid sequence. So here's a strand that could represent any generic protein. Those R groups represent the amino acid side chains. So you kind of break them down one chunk at a time and just say they're a string of amino acids all linearized down the line. We can refer to the N-terminal and the C-terminal side because there's an amino group and a carboxy group on each end. And ultimately, that's how these amino acids were linked together. So you look at the amino group of one side, the carboxy of another, and then you remove water in the chemical reaction that brings these two things together. Of course, enzymes would mediate that reaction, and we could go in reverse if we wanted to. Just throw some acid at a protein, it would go in reverse. Now, a protein is much more than the sum of its parts. So when it comes to the protein sequence, that defines, in the primary sense, what a protein is. But that primary sequence can then adopt a higher order structure. So the protein can fold and it takes on a specific shape where that shape decides exactly what the function of the protein is. So proteins are three-dimensional structures, and that shape, that folding that they take on, plays a lot of a role in deciding what kind of protein or what specifically it does. And fundamentally, what decides if a protein is folded this way or that way, it comes down to the property of the amino acids, the polar ones and the non-polar ones. In general, a protein will be in a water environment, so the hydrophilic components will be pointing out, and the hydrophobic components will be pointing in. But of course, not all proteins are embedded in an aqueous environment. Some of them embed themselves in the more fatty parts of our body, specifically the cell membranes. And these cell membranes are lipid bilayers. So in this case, it's pretty much the opposite. So membrane proteins in particular have like a lot of hydrophobicity to them, which actually makes it a lot more difficult to be able to study them. So it's hard to even dissolve these proteins to begin with. So that's just a taste, just a sense of where it is that we're going to be going. We still have to talk about how mass spectrometry studies proteins, how we get the amino acid sequence. And even before that, we have to understand the other components. So mass spectrometry doesn't do it alone. But from here, what I'm saying is that genome sequencing has its strong place. It's like an indispensable tool in science today. But at the same time, it doesn't necessarily give you all of the answers. So proteomics is a complementary tool that allows you to study proteins directly. It's kind of a fun story. It's what I'm doing every day in my lab. Well, whenever the lab's open, whenever I can get there. But we'll talk more about that in the next video.
Okay, genes within population. We will look at genetic variation in natural populations and consider the evolutionary forces that cause allele frequencies in natural populations to change. So remember, what are alleles? Alleles are versions of genes. So there are times when you have changes in the allele frequencies in different populations. There are times when you have certain alleles that are more common in certain populations than others, all right? Darwin proposed natural selection as a mechanism of evolution. We will understand what natural selection is, and we will learn that natural selection is not the only mechanism of evolution. One of the first ones we will talk about is mutations. Mutation has a lot to do with evolution, all right? So we will look at all the 5 key components that contribute towards evolution. One way to monitor how populations change through time is to look at changes in the frequencies of alleles of one gene from one generation to the next. So you can study different populations, and you can see, all right, the frequencies of this particular gene was X amount, but 10 years down the road, the frequency of that population, of that allele, the frequency of that allele has changed. What caused that change in the frequency of that particular allele? All right, is it natural selection? Is it mutation? Or is it something else? Natural selection, by favoring individuals with certain alleles, can lead to change in such allele frequencies, but it is not the only process that can do so. All right, so what it is saying is that natural selection can lead to a change in the frequencies of alleles within a population, but natural selection is not the only one. You have others as well, mutations, genetic drift, and so on, okay? Allele frequencies can also change when mutations occur repeatedly. So changing one allele to another, and when migrants bring alleles into a population. So there we go. That's another factor that can cause frequencies of alleles to change. Migration. So it happens in human population, and it happens in animals, and it happens all around. So when you have migration, maybe a particular species of birds come from somewhere else and come in this area, they bring their own unique alleles with them. And when they start to interbreed, then you can see whereby you can have changes in terms of the frequency of alleles within a population. All right. You look at the population of Poland, for example, is the gene that is responsible for melanin very frequent in that population? It's not. But if you have migration, whereby you have some people of dark complexion start going there and intermingle and mating with these people from Poland, then you can see whereby over a period of time there will be changes with respect to the frequencies of certain alleles. Okay. Evolution can result from any process that causes a change in the genetic composition of a population. We cannot talk about evolution, therefore, without also considering population genetics, which is the study of the properties of genes in populations. So population genetics has to do with that study of genes and their frequencies within populations, all right? So this chart outlines the 5 agents of evolutionary changes, or the 5 what would cause evolutionary changes. So the first one here is mutation. We know what mutation is, all right? So individual mutations occur so rarely that mutation alone usually does not change allele frequencies much. We will look at mutations and go in depth in mutations. We know about the different types of mutations. We will study them. All right. Thereafter, we will move on to gene flow. So individuals or gametes move from one population to another. All right. So we're talking about migration. Organisms migrate to different areas, and then they start to intermingle with other organisms in that area, and they cause a change, a shift in the allele frequencies within that population. Okay, so gene flow, moving organisms from one area to the next. So migration as well, that is what gene flow is all about. Random, non-random mating. This occurs especially in self-fertilization organisms. So inbreeding is the most common form, and it does not alter allele frequency, but reduces the proportion of heterozygotes. So when you have self-fertilization taking place, it more promotes homozygosity than heterozygosity because you're using the same genes. When you have crossbreeding, then you're getting genes from one organism and mixing it with another organism, and so that would stimulate more heterozygosity because you're having combinations of genes from 2 different sources. All right? Genetic drift. So this is the random fluctuation in allele frequencies that increases as population size decreases. You get genetic drift when you have migration as well. Organisms moving from one area and they go and populate a different area. All right. And so you can have a shift in terms of the genetics in a particular area. Selection. All right, so you have natural selection and you have artificial selection, and then natural selection can also be broken down as well into sexual selection and ecological selection. A video will clarify all of that. But selection, this is the only agent that produces adaptive evolutionary changes. Adaptive evolutionary changes. If you look at that diagram, what is happening there? This picture. You notice that the bird is eating these caterpillars. It will eat the ones that have that bright yellow color. So what actually takes place over a long period of time is that these caterpillars adapt. So they change color and they turn green. so that they can be camouflaged with the leaves that they live on, so that the birds would not be able to identify them. So these adaptive evolutionary changes is a means of survival. They do this in order to survive. All right, so these are natural selection. Artificial selection, on the other hand, is when you have mating occurring and it is directed by human beings. So, for example, we're going to breed dogs. I want to breed a German Shepherd and I want to breed a Rottweiler. Well, we're talking about two completely different sets of genes. So I want to see, well, the alleles from there and the alleles from there, if I mix them together, what will I get? All right, so that is artificial selection. When you, human beings, intervene in terms of selecting alleles that will move on to the next generation. In the case of natural selection, that is something done naturally by the environment. Okay? I will show you guys this other video that will summarize these 5 agents of evolutionary change.
All right, now we're going to do agents of evolutionary change. And don't worry if you're more interested in those homologous structures, vestigial structures, the fossils, we're going to go back to that. But right now what I want to do is I want to kind of connect it more to genetics, since that's what we just finished, and this like more microevolution. So dealing with the smaller scale changes and talking really about that natural selection, because that's going to be one of the keys for this unit. All right, so today what we're going to talk about is agents of evolutionary change, or in this video. You should be watching 2 videos today. The agents of evolutionary change. So we've got 5 main things we're going to talk about. So one is mutation. Now, mutation is any change in the DNA sequence. Now, what I wanted to remind you is that when we have a change in the DNA sequence, that doesn't always change the amino acid sequence. It doesn't always change the protein. Even if it does, that doesn't always change the structure or the function, but it can. So if we have a change in the DNA sequence that then does change the amino acid sequence, remember that's going to be more likely if it happens during the first and second amino acids in our codon, because remember our codon would be something like this. And this last one can often be a swing. So if you look at your codon chart, a lot of times you might have, and this is a bad example because this would be really a UG, and that would be the start codon. But a lot of times what we have is we have things like, let's do CCC. CCC is probably the same as CCG, CCA, and CCU. Usually we have this last one will swing, and at least 2 of them will be the exact same amino acid. And I'm not sure if that's correct. I can't look it up on a codon chart right this second to make sure which amino acids are coming from this codon, but we will usually see that we don't get a change in amino acid if we're changing that last nucleotide in the triplet. If we get an amino acid change, that might change the protein. It could change it dramatically. Remember, sometimes if we change to a stop codon, that can change the protein by making you have only half a protein. If we change it from one amino acid to another, that might change the structure. It depends on what amino acid is changing. So if we're going from a like acidic amino acid to a basic amino acid, that will definitely change our structure. And a lot of times structure equals function. If we change our structure, we're going to change the function. Now, if all of those things change, we will often see this change in phenotype and therefore fitness. But again, there's question marks each step because it depends on where that mutation is, whether each thing is going to happen. All right, next I want to talk about gene flow. Gene flow is a concept where we talk about movement of individuals into and out of populations, and that this can cause the population's genes to change. So when we're looking at a population level, because remember, evolution does not happen in an individual, evolution happens in a population. Now, this can happen a lot with things like seed and pollen distribution, because seeds and pollen are going to be distributed basically randomly, especially seeds that are distributed by wind. Animals can migrate. So you might have subpopulations that have very different allele frequencies. So for example, this subpopulation has all recessive alleles. This subpopulation has all dominant alleles. If one of these birds goes over to this population, this causes this genetic mixing. This adds in new alleles into this more dominant population. It reduces differences. So if we have gene flow, it reduces differences between two populations and makes them more similar. So gene flow is the exchange of genes between two populations. Next is non-random mating or mate choice. So I put two pictures here of examples. Birds are the best example. We almost always use birds as examples in biology because they display their mate choice very vibrantly. The males tend to be bright colored, the females tend to be more drab, and then they can be choosy. So this example with the peacock. The peacock has all these bright feathers, the male does anyway, and the female is more like brown, and it can pick what male peacock it wants to mate with. Cardinals, which I've been seeing out and about lately, the more red the cardinal, the more desirable he is from a mating standpoint. One of my friends in grad school did a study on it, which she called having good genes. Good in quotes there, that, the red cardinals seemed to have the better genes because they were sought after by the female cardinals. This is an example that you may not have seen too much before. This is an example of an aquatic creature. That makes these patterns in the sand. And it makes these patterns in the sand in order to attract a mate. So it's almost like sand art. So these fish, trying to remember what kind of fish is it. It's the pufferfish. That's what I thought. The pufferfish. I just had to Google to double check. So the pufferfish will actually make these patterns in the sand as part of the mating ritual in order to attract the female. So that is another example of non-random mating. So the female is choosing a mate based on this behavior, which means that her, pufferfish pups will be more likely to exhibit that same behavior. Genetic drift is another example. This happens due to random sampling. We have two examples that we like to talk about, the founder effect or the bottleneck. A founder effect is when we have a small group that starts a new colony. So for example, we have a red bird or a couple red birds that fly to this island. They start a new colony. Because this colony was started by only red birds, it doesn't have those yellow alleles. The other one is a bottleneck. And in a bottleneck, let's get this out of the way. In a bottleneck, you have some factor that comes through and reduces the population to a small number, and then the population expands again. But the key is that it is random. Whoops. So the key here is randomness. So we don't have something that comes in and removes all the red. It's randomly coming through and just taking a whole bunch. So if you do that, if that bottleneck happens, you see we went from 20% of each color down to we still have 20% green, we still have 20% red, but we only have 0.14 pink, 0.06 yellow, and 0.4 blue. So by this random thing, we have changed our frequencies. And this is, again, removing numbers until we get to this 10th generation. And in this case, we're just removing individuals to see how we can change our percent. So by this time, we have 0.2 blue, so we're back to 0.2 for blue. But yellow and pink have both gone extinct, and red has gotten to be the most in the population. And this is all randomness. Here's another example of that bottleneck. So this is why we call it a bottleneck. You have this parent population. Imagine it's in a bottle and you're pouring it out, and only a couple can get through the neck of the bottle at a time. So there's some disaster. It means that most of what's in that bottle will not survive to the next generation. And so some of the alleles are lost from the gene pool, but it's not due to fitness. It's due to randomness. So this red allele is just not very common, and it's completely lost to the next generation. Also, the yellow is about as common as the blue, but as you can see here, because of just the randomness of the bottleneck, we're left with mostly blue individuals. So this narrows our gene pool. And remember, that is part of your genetic drift. Cheetahs are a good example. All cheetahs share a small number of alleles, less than 1% diversity. It's as if all cheetahs are basically identical twins. We think this is because of two separate bottlenecks, 10,000 years ago at the end of the Ice Age, and then the last about 100 years ago due to poaching and loss of habitat. So based on their genetic information, based on looking at different mutations, we're pretty sure about when those two bottlenecks would have occurred. And then last is natural selection. We've talked about natural selection a lot. The less fit animals are removed, in this case, eaten by this bird who was hungry. It can see the blue caterpillar more easily on the green leaves than the green caterpillar, and so it can eat those. You can also have differential survival due to environmental conditions. So things like climate change, food source availability, predators, parasites, diseases, all of those things can cause differential survival based on the genetic makeup of the individuals. And then also toxins. So like toxins present in food items or toxins present in the environment in other ways. So like this could be people adding them. The combination of alleles that provides fitness will increase in the population, and then you will be left with those fit individuals. All right, so for practice, give an example of genetic drift that is not an example.
All right, gene mutations. So we will focus on the first agent of evolutionary change, which is mutation. Gene mutations. A mutation is any detectable and heritable change in the genetic material not caused by genetic recombination. So remember that when you have genetic combination, for example, mating, sexual reproduction, you have a combination of genes and then you end up with a progeny or an offspring that has a unique combination of gene. So there's a change in the overall genome of these progeny. So that is not considered a mutation. A mutation would be a change in the genetic material of a particular individual. A mutant cell gives rise only to somatic cells. This type of mutation is called somatic mutations. These are not passed on to succeeding generations. However, mutations in the germline of sexually producing organisms may be transmitted by the gametes to the next generation. Such mutations are called the germline mutations. These result from unnatural change that affects DNA's chemical or physical constitution, its replication, its phenotypic function, or the sequence of one or more DNA base pairs. So we're talking about adding base pairs, deleted base pairs, substituted or put in reverse order, inverted at 180 degrees, or transposed to new positions. Types of mutations. A mutation can occur at the level of the chromosomes or at the level of the genes. A change in the organization of a chromosome is called a chromosomal aberration or chromosomal mutation. All right? So remember, when we talk about genome mutation, we're talking about a change in the genome that affects the chromosome number. All right? Chromosomal aberrations, we're talking about these four primarily: deletions, duplications, inversion, and translocation. All right, so these are the four main types of chromosomal mutations. So these affect chromosomes. So type of chromosome changes, you have chromosome aberrations or variations from the wild type condition in either chromosome structure or chromosome number. So So variations in chromosome structure. This type of chromosome aberration involves changes in parts of individual chromosomes rather than the whole chromosome or sets of chromosomes in a genome. Like I mentioned, there are these 4 types: deletions, duplications, inversions, and translocation. In duplications, both involve a change in the amount of genetic material on a chromosome. So you have deletions and duplication will result in a change in the amount of the genetic material. That is referred to as the C value. So the C value refers to the amount of genetic material in a genome. When you have deletions, the C value is affected. When you have duplication, the C value is affected. All right? Referring to the amount of genetic material that is changed in deletions or duplications. On the other hand, in inversions, it involves a change in the arrangement of a chromosome segment. So a piece of chromosome is inverted at 180 degrees. So it is inverted at 180 degrees, and hence the C value, or the amount of genetic material, does not change, because it's one segment of DNA that is inverted 180 degrees. Translocation involves a change in the location of a chromosome segment. So you can have a piece of chromosome that breaks off from this one chromosome and reattaches itself to a different chromosome. That is translocation. Okay? Let's we'll stop there for today, and on Thursday we will discuss a little bit more about deletions and duplications, inversions, and translocation. We'll just go a little bit more in details in each one of these different types of chromosomal mutations. All right? So any questions before we leave?
Sir, this PowerPoint hasn't been posted as yet, right?
No, it's not posted as yet, but I'll certainly do so.